Cell differentiation. How do cells become specialized for different functions? During the development of an organism, cells differentiate into many types of cells. All organisms start life as just one cell. Most multicellular organisms pass through an early stage of development called an embryo, which gradually develops into an adult organism. During development, an organism's cells become more differentiated and specialized for particular functions. For example, a plant has specialized cells in its roots, stems, and leaves, cells that transport material, cells that store sugar, cells that carry out photosynthesis, and more. The process by which cells become specialized is known as differentiation. During development, cells differentiate into many different types and become specialized to perform certain tasks. Differentiated cells carry out the jobs that multicellular organisms need to stay alive in homeostasis. Cell differentiation in mammals is controlled by a number of interacting factors in the embryo. Adult cells generally reach a point at which their differentiation is completed, and they no longer become other types of cells. What are stem cells? The unspecialized cells from which differentiated cells develop are known as stem cells. One of the most important questions in biology is how all of the differentiated cell types of the body are formed from just one single cell. Biologists say that such a cell is totipotent, literally able to form any of the tissues of the body. Only the fertilized egg and the eggs produced by the first few cell divisions of embryonic development are truly totipotent. After about 4 days of development, a human embryo forms into a blastocyst, a hollow ball of cells with a cluster of cells inside it known as the inner cell mass. The cells of the inner cell mass are said to be pluripotent, which means that they are capable of developing into many, but not all, of the body's cell types. Stem cells are unspecialized cells from which differentiated cells develop. There are two types of stem cells, the embryonic and the adult stem cells. The embryonic stem cells in a culture are specialized eventually to become things like macrophage, smooth muscle cells, fat cells, neurons, and others. In this embryonic stage, the blastocysts include the inner cell mass. Contained inside the blastocyst is the inner cell mass, and these are pluripotent. Embryonic stem cells are found in the inner cell mass of the early embryo. Embryonic stem cells are pluripotent. Researchers have grown stem cells isolated from human embryos in culture. Their experiments confirm that the embryonic stem cells have the capacity to produce all cell types in the human body. Adult organisms contain some types of stem cells. Adult stem cells are multipotent. They can produce many types of differentiated cells. Adult stem cells of a given organ or tissue typically produce only the types of cells that are unique to that tissue. What are some possible benefits and issues associated with stem cell research? Stem cells offer the potential benefit of using undifferentiated cells to repair or replace badly damaged cells and tissue in the host. Stem cell research may lead to new ways to repair the cellular damage that results from heart attack, stroke, or spinal cord injuries. One example is the approach to reversing the heart attack damage illustrated below, where stem cells are filtered from the bone marrow removed from the patient's hip. And the stem cells are then injected into the heart's damaged area. The environment of the heart stimulates injected stem cells to differentiate into new heart muscle cells. Human embryonic stem cell research is controversial because one procedure harvests cells from the blastocysts or earlier that would grow into a human if incubated in the mother's womb. These cells are instead used to aid the health of another human. Arguments for it and against it both involve ethical issues of life and death. Most techniques for harvesting or gathering embryonic stem cells cause destruction of the embryo. Government funding for embryonic stem cell research is an important political issue. Groups seeking to protect live embryos oppose such research as unethical. Other groups support this research as essential to saving human lives and so view it as unethical to restrict research.